Ok, grazie a tutti i presenti. Forse questo è il seminario che è riuscito a riempire in aula, in tutto il dipartimento, non è mai capitato, per cui siamo molto contenti del fatto che avesse suscitato tutto questo interesse. Il giornale piccolo mi ha chiamato la settimana scorsa e diceva: ma tu sai questa storia? Chi è che viene dal sangue, eccetera. Abbiamo la dottoressa Marcela Uliano, che è attualmente Senior Bioinformatician al Wellcome Trust Sanger Institute, che, se non conoscete, è una delle più prestigiose istituzioni mondiali per la ricerca sui genomi, le scienze molecolari. Il genoma umano del 2001 è uscito dopo 20 anni di studi, di tentativi, grazie soprattutto a questo centro di ricerca, non solo, ma l'iniziativa pubblica sicuramente è uscita da quell'istituto lì. Marcela è una nostra, diciamo, conoscenza. Abbiamo ricordato oggi che è stata qua 13 anni fa, quando faceva il suo master all'Università Federale di Rio de Janeiro. Ha preso un PhD a Rio, giusto? Con il qua presente il nostro caro amico e collega professor Mauro Rebello. Poi è stata a Berlino all'Istituto di Biodiversità, dove ha fatto il suo postdoc, e adesso giustamente ha superato tutti noi e si è insediata al Sanger Institute e lavora per un progetto importantissimo, prestigiosissimo, che è questo progetto del Tree of Life. Ok? Adesso non vi spiegherò cos'è il Tree of Life perché non vi voglio anticipare nulla, per cui ci godremo il seminario di Marcella assieme. Il seminario è in lingua inglese, naturalmente. Marcella capisce bene l'italiano, però non si sente ancora, diciamo, sicura di parlare in italiano. Giustamente questo è un seminario scientifico, per cui parlerà in inglese, durerà circa 45 minuti, dopo ci sarà una sessione di Q&A e vi invitiamo a restare, insomma, fino alla fine dell'evento. Grazie ancora, lascio la parola a Marcella.
Allora, ragazzi, ma se avete domande, poi potete fare in italiano. Io capisco, ma non posso parlare velocemente o fluentemente, no? Mi dispiace. Ok, passo all'inglese adesso. Ok, e facciamo così piano piano. Hi, everyone. Okay, so Francesco has introduced me. I have this slide here, which just basically tells everything that he has said now. So I did my master's and my PhD in Brazil. Then I spent some time in Berlin. Then I got this position at the Wellcome Sanger Institute. I'm also affiliated to the University of Cambridge, and I'm also an associate professor at Nord University in Norway, but my main base is Cambridge. So I spent most of my time in the UK, based at the Wellcome Sanger Institute. Right, so to talk about me and my research, I can tell you that I have two main lines of research. One is software development to automate high-quality genome assembly, because today I will talk to you about this project, the Darwin Tree of Life project, where we are sequencing 70,000 species that live in Britain and Ireland. So we cannot do one genome, one person all the time. So we need to automate algorithms to do genome assembly. But I also do comparative genomics. So I take these genomes and then I study biology and evolution using these genomes. And today, I'm not going to go into a specific story. I will present you the Darwin Tree of Life project. But I just want to mention this work with these animals called Sicilians very quickly for us to introduce this talk. So just for you to understand how we are using genomes to study biology. So snakes are reptiles, right? And they don't have any limbs, as we know. So a few years ago, people found a specific region of the snake genome that they thought it was related to the fact that they don't have limbs, right? Because lizards, which are also reptiles, have limbs. So they compared the lizard's genomes with the snake genomes and found this region of interest. And then they took the sequence and put it in a mouse. And then basically the mouse, so this image is just to show you a comparison between lizards, genomes, and snakes. So this is in the context of a phylogenetic tree, where you basically take all the sequences of DNA and you try to find similarities between different species. So they took this sequence that they found and they put it in a mouse, and that happened. So the mouse came out as a serpentized phenotype, right? Which was showing us that was very likely that that region of the snake genome was related to limblessness or not having limbs. Then a few years ago, myself and some colleagues, we sequenced Sicilian genomes, which are another type of species. Okay, they are amphibians. They are not reptiles. And the Sicilian genomes also are limbless, right? They don't have any limbs. And frogs, which are very related to Sicilians, have limbs. So basically looking at the Sicilian genome and having the specific genotype of the snake, we compare the sequences of these genomes, the Sicilians and the snakes, and we basically see that the Sicilians, which are here, also don't have this very dark blue sequence, which is this ZRS enhancer. It's also absent in the Sicilian genomes. So this is in biology what we call convergent evolution, which is basically the same molecular mechanism that exists that gives a specific phenotype in distant related species, right? So the same happens in amphibians and in reptiles. And we can find this basically because we have sequences to compare, right? Genomes to compare. So because in biology, nothing makes sense except in the light of evolution. And for genomics, it's the same thing. So nothing in genomics makes sense if not in the light of comparative genomics. So you always need to look at sequences from other species to try to make sense of the DNA material. And this is the reason why we are developing this project to sequence all life on Earth. So we are sequencing reference genomes for all the species that exist in this planet. So I will show you how. So the project that I work on is this called Darwin Tree of Life project. So we have developed sequencing technologies enough now that a few years ago, scientists got together and said, okay, now we have the technology that is advanced enough to sequence reference genomes for a whole life, and then they will stand the test of science in time, right? Because in the past, we were sequencing some genomes where we always needed to make them again because the technologies were evolving, and then we were getting like longer sequences for the genomes. But now we know the technology is advanced enough. Even though there will always be technology improvement, the references that we are producing now are basically complete, okay? So then lots of scientists gather in different consortia, okay? So there are regional consortia, such as the Darwin Tree of Life. So we are sequencing everything that lives in and around Britain and Ireland. And then we have consortia that are taxa related, like the Vertebrate Genomes Project, which wants to sequence all vertebrates. So the idea of these different consortia is to gather references for the use of all, okay? And all of these different consortia are under the umbrella of the Earth BioGenomeProject. So, yeah, so the Earth BioGenomeProject is basically trying to organize everyone around the world to sequence, at the end of the day, everything. So I want to show you this video from our project because it's nicer than me just talking all the time. So I hope that it's a 3 minutes video, and I hope it will play. Here. Yes, and I need to share, I think. So I need to stop sharing this and share that, which is this. Share. And now I will press play. Let's see if it...
I'm head of the Tree of Life program at the Wellcome Sanger Institute. Our goal is to sequence the genomes of all the fungi, plants, animals, and protists on this planet, both to understand how they've evolved, how they work, but also to use that information to conserve biodiversity and to promote human health. The way we're approaching this huge project of sequencing everything is by starting on a number of named projects. So one of these projects is the Darwin Tree of Life project. That project is to sequence everything that lives in, on, and around the islands of Britain and Ireland. This is a collaboration with people across the UK in museums, botanic gardens, and other institutes to collect specimens for us. So the botanists at botanic gardens know where all the species of plants are. The marine biologists know where to go on the beach and where to dive in the cold waters of Britain and Ireland. And the Naturalist Museum and their army of collectors know where to go to find all the species that we need to sequence.
I'm Ines Janishchak, and I'm the sampling coordinator for the Darwin Tree of Life project at the Natural History Museum. We're currently in Millport, and the idea is we want to sample a wide range of locations around Millport for marine species. Our main focus is invertebrates. So we're basically trying to get a snapshot of what's here so we can add it to the project and increase our species list. In the lab at the moment, we're actually just going through every single individual tray and we're picking out all the things that look interesting. We have an ID team who are going to ID it to the best of their ability. We then process them at the Natural History Museum, and then they are sent to Sanger for whole genome sequencing if things go well.
I'm Graham Oatley. I'm an advanced research assistant in the Tree of Life core lab. We work in a laboratory and taking the samples that we get from partners, extract DNA and RNA from them, and then pass them over to the sequencing team. At the moment, there's no one-size-fits-all extraction protocol. So the key challenge is to try and develop different extraction protocols for the different organisms that we're getting in to get the high-quality genomes that we're aiming to sequence for the Darwin Tree of Life.
I am Marcela Uliano da Silva, and I am a senior bioinformatician in the Darwin Tree of Life project. So after the samples being sequenced, it comes to our team, and we assemble the genomes of all of the species that come through. So assembly is basically the process of taking the shorter DNA sequences and finding overlaps between them with the help of computer software to build up ideally chromosomes. So genome assembly is basically like assembling a jigsaw puzzle, which varies in shapes and sizes.
I'm Charlotte. I'm a PhD student in the Darwin Tree of Life project, and I'm studying the evolution of butterflies and moths. Moths and butterflies are extraordinarily diverse. In fact, they are 10% of all described species. And in the past, studies that have looked at genomic data have been limited to perhaps 3 species. So you can say what's true in those. But as part of the Darwin Tree of Life, we've been able to sequence hundreds of these Lepidopteran species. And the interesting thing that we've found so far is that Lepidopterans are kind of a story of 2 sides. On the one hand, most species have undergone very little change. But in a few cases, they've undergone dramatic changes. They've stuck together bits of their chromosomes and then split them up again, which means their overall genome looks very different. And we're really interested in what has allowed them to avoid this constraint that the rest of Lepidopterans face.
My job here at the Tree of Life is my dream job. Ever since I've been a scientist, I've wanted to understand organisms at the genomic level. And every time I looked at a new organism, I wanted to look at more. And so suddenly here I am with some amazing colleagues able to do that, to sequence at scale across biodiversity. And that to me opens huge vistas. Rather than understanding how this one species does what it does, we've got to understand how all species do what they do. And I believe that this is going to change the way we do biology forever.
Okay, and then what did it kidding. So, but you know, basically it. And I will explain you now a little bit of the details of the project. Yeah. Right. So yeah. So I guess I hope that the system story justifies a little bit and makes you understand why we need lots of different genomes, right? And it's interesting because we have seen a revolution in genomics in the last 20 years, and we learned a lot about molecular biology. But if you consider the different types of adaptations and ways that life exists in this planet, we are very vertebrate and human-centric. So we look at mammalian genomes a lot, and there is a lot of other types of ways that life is adapted that are important for us to understand. My main interest is the biology itself, but it's also very good for biotechnology application, right? So the ways that the thing catalyzes different reactions and so on, and how life exists in this planet. So we don't know much of it yet. So having all of the genomes, having lots of the genomes, we are able to find the conserved blocks across the tree of life. We are able to find the where the symphonies break in specific adaptations of radiations of several different species. So how these specific adaptations are done molecularly, and how the species are going to cope with, for example, a change in climate. It also is a very nice project to reference genomes for conservation biology. Conservation biology doesn't need genomes all the time, but many times it's useful to see how the population is doing in terms of diversity in the process. So this is not a project that we are starting here, and then 10 years later, we have all the genomes, and then the science starts. As we develop the project, lots of science is being done, and I hope I can show you this. We are advancing not only the understanding of molecular biology, but also several methods to advance the field of genomics. So it's Britain and Ireland. So this project is heavily funded by the Wellcome Trust, right, which is a British fund. But it is interesting to sequence regionally Britain and Ireland, because in this island, we have species that represent 40% of all described families on Earth, which gives us also a good phylogenetic depth for us to understand life and do these comparisons that I was talking about. Our project then wants to sequence 70,000 species of eukaryotic organisms, so organisms that have the genome in the nucleus of the cell, not bacteria, right? So plants, fungi, animals. And it's a partnership between lots of different institutes, okay? So the main sequencing is done at the Wellcome Sanger Institute, but we have partnership with the Natural History Museum, University of Cambridge and Oxford, heavily funded by the Wellcome Trust. So we are many different people, like taxonomists, field scientists, wet lab scientists, bioinformaticians, and evolutionary biologists. So it's a nice, very nice project in the sense that we really empowers and employs biologists in very different layers of the chain, right? It's not only based on tech at the end of the project. So our goal is to generate high quality chromosomally complete reference genomes for all named eukaryotic species that are native to Britain and Ireland, to annotate these genomes and to release them openly for the use of all. So we produce the genomes, and we also produce RNA-seq data, which is the expressed part of the genome, to do annotation, to find the genes on these genomes. So I will show you a little bit about our so-called genome engine. So it's basically this path here. I will show this slide briefly, and then I will go into more detail. So basically, the first part of the project is to acquire the samples in the field, right? And then also, we need to record the metadata. So imagine it's 70,000 species, and sometimes we collect more than one specimen for each species. And then when we go to the lab, we also separate in tissues. So there is always like more than one sample for each thing. So it's like 70,000 times, like many times, which really means you need to have metadata stored, right? So there is this tool called POPO, which stores all the metadata for our collections. Then the specimens are morphologically identified by taxonomists, and then it comes to the Sanger. So, and then the DNA and the RNA are extracted in the lab. And again, they have like a barcode and lots of metadata recording for the specimen when it's in the lab. As soon as things go to the sequencing machine, then it comes to our team. Well, the team that I used to be part until the end of last year, now I changed teams, but I was in the assembly team until December. And again, imagine, so we also sequence more than one sequencing technology for each specimen that we sequence. So it's PacBio and then Hi-C. And what happens is that you can have sample swaps a lot, right? Because it's like it's only human. You have one scientist extracting each specimen that comes to the lab. So in the assembly team, we need to develop like a method to also check that the DNA is from the same individual, so PacBio and Hi-C. So I developed a protocol at the time when the project started to like check the IDs of each sample. After everything is checked, then it goes to assembly. So we basically take these smaller pieces of DNA, finds overlaps between the DNAs and build up ideally chromosomes, so longer stretches of DNA, no? After this is done automatically by our machines, but with lots of bioinformaticians checking the process, then it goes to this manual process, which is called genome curation. There is a lot that the machines cannot do still with lots of repeats on the genomes. Genomes are very complicated to assemble, to build up. So then curators go and look at and change a structure of chromosomes manually. As soon as this is done and like done by the curation team, then it goes for annotation to another institute, which is the EBI, the European Bioinformatics Institute, which is also located in the same campus as us, as the Sanger Institute. And they are our partners there, do genome annotation for this, for all of the genomes that come through. And they are then openly released in our portal and also in all of the normal INSDC public databases for DNA sequence, such as NCBI or ENA, okay? And then after they are publicly available, they are always available for the use of everyone, always. And if there is something that is in the process of sequencing that people would like to know about, they can always email us and then we say where the status of that sequencing is. But as soon as they are available, we also produce a genome node. So we are automating this process at the moment as well, which just basically describes all of the quality metrics for that genome, and then people can cite that genome node when they are using that genome for research. So in a nutshell, it's basically that. This is like people that do the very nice part of the work, right? So just to show some images of our partners in the field. So we have the Whiteham Woods, where we collected lots of butterflies and moths, especially during COVID, because there was restrictions on where you could go for collection. And this is someone just in the Royal Botanical Gardens of Edinburgh collecting bryophytes. If you want to know more about what's going on in the field, you can always check the blog as well. So this presentation is public. I have a link for it. If you like later to like take notes of or everything, I have it public. So you can just have it. So at the center of our sequencing initiative at the Sanger is PacBio. So PacBio is a technology that produces long stretches of DNA sequences, okay? Because DNAs of eukaryotic species are made of repeats, palindrome sequences. So we need to sequence very long to be able to, we say, span these repeats and really get an integral sequence reference for the chromosomes of the genomes, okay? So we use what we call long-read sequencing together with long-range sequencing, which is this other type of technology called Hi-C, to do with scaffolding, to basically take all of these conjugates and bring them to the chromosome level, like scaffolding them across centromeres. I will show you slides on this soon. And so as much as we are automating the assembly part, the lab part is always a challenge for projects like this because it really requires one scientist, one sample. At some point, right, there are robots that do some parts of the processing of the library, but at the beginning, there is always someone that takes a tube with a tissue from one specimen, no? And we will likely to do that 70,000 times or more. And these people, which is really nice, the core lab, they are basically also developing lots of protocols because we are sequencing across the tree of life. So we are very used to protocols for human samples, mouse samples, and then these protocols don't work for fungi and plants and other things, no? So these guys are actively developing protocols, and they have a protocols IO with lots of different protocols. They are also on Slack because lots of people email me asking, I have a fun medium extracting DNA. How do I do it? I always point it to them because they are the experts and all this information is out there. And they are also very accessible and available to talk about sample extraction. Because to assemble a genome, you need to take from the nucleus of the cell the most integral DNA you can. But the process of extracting DNA breaks the DNA, right? So they basically just optimize protocols to take it as long as possible. So then we can sequence as long as possible and makes the life of the bioinformatician easier in the assembly process. Right, so assembly. So this was the team I was part of until December. Now I'm in Mark Blackster's team, which was the guy that appeared in the in the video, but I will show you a little bit what we do in terms of how we assemble these genomes at the Sanger. So this is our assembly pipeline. So very quickly, this is the type of sequencing we do, which is PacBio. So if you look here, this sequence here is a piece of DNA, okay, double strand. And then you basically put like adapters at the ends of this DNA sequence. And then you open these two strands and you sequence, like do many passes of it many times. So you sequence it many times, the first strand, the second one, the first strand, the second one. And then you create a consensus of a long read that has high quality per base of DNA sequenced as well. So this type of technology is really nice because if you have a diploid species, which is like humans, a species that has 50% of the genetic material from the mom and 50% from the dad, when we sequence very long stretches of DNA, we can find the differences between the mom and dad haplotype really well. And we can basically separate both haplotypes and have two genomes, the genome of the mom and the genome of the father, which is really nice. Because in the past, we used to like merge this information, and then we had genetic variation from both in one reference, and that caused a lot of problems for genome sequencing. But now we can separate those haplotypes, which is really nice. Then we use a tool called purging to do the separation. For a while, we had a step which was polishing, we used to call, when we use short read sequencing to increase the quality per base of the longer reads, but PacBio Hi-Fi is advanced enough that we don't need this anymore. So for all the mitochondria, we have the software which is called Mito Hi-Fi, which also assembles the mitochondrial genome of the species. And then once you have very long stretches of DNA already sequenced, then we use the 3D structure of the chromatin. So I don't have time to explain exactly how the technique works, but we basically take the 3D information of the chromosomes. So the chromosomes in a somatic cell, they have contacts with itself like this. So here, each color is one chromosome. And then we basically freeze this information and then we map read sequence like that to create like longer stretches of DNA. So for the human genome, in biology, we have the centromeres, right? So we have telomeres, lots of repeats at the end of the chromosomes. Then we have sequences with lots of genes. And then we call chromosome arms, right? For species that have chromosome arms. And then in the middle, we have the centromere, which is the part of the DNA that when DNA is condensed to separate, the proteins go there and separate the two arms of the two copies of the chromosome, no? So the centromere is a region that is very repetitive and is very difficult to sequence across it. But then with techniques like Hi-C, we can get the two arms of the chromosome together and we can reconstruct the final structure of the chromosome. So this gives us what we call genomes out of the box. So I will explain you what this means. So here in this diagonal, so we have 4 different species, right? And then each of these squares, for that species here, we have a map. So this map here, each of these squares is a chromosome. So trust me, it's just a visual way of looking at DNA sequences, okay? So the intensity of the red means that all of the Hi-C reads are mapping only to that chromosome. So this shows us correctness of structure, okay? So I don't have any off-diagonal red signals. Here, if you look, this small chromosome here, it has some Hi-C reads that map to it and then map to a smaller thing. So there is a tiny error there, which is the kind of things that the curators go and check manually. But for Lepidoptera, which are butterflies and moths, our pipeline works really well. But then we have things like this. So lots of species still need a lot of manual curation. So here in black are lots of tiny pieces of DNA that don't have any Hi-C signal, first of all, so we don't know where to place them. The other things are very high scale that I can show you is that when you have a chromosome here, but you have signals that like also match to a tiny thing there, usually it means that you need to take this chromosome and put it there because they belong together, but automatically the algorithms weren't able to join this DNA. So then the curators do that manually many times until they get to something like this, which is like much better. And in this case, it's like a a brutal example that they needed to do more than 1,000 joins manually and 372 breaks. So, and to get to chromosome level. So now that we have more than 1,000 species sequence, we are training machines to do machine learning to try to do that automatically, but this is still a very manual process at the moment. So again, this is another example, okay? So basically automatically there was a lot of things that were up that should be together, they were not. And then the curators just need to do that by hand.
Then the size of the squares on the right side means that the chromosomes increase in size also.
Yes,
So
yes.
you have a lot of small squares there, and then
They join together and then make.
join together a lot of DNA that was dispersed.
Yeah, because it's just basically like ATCC. Let's say this guy is ATC, and then this other one is GGG, no? GGG. And then at the end, they will be together, right? So it's a larger sequence. Yeah, so the size of the square is representative of the sequence of DNA, right? So if we consider that this is chromosome 1, this would be like base pair 1 of the sequence, and then in the diagonal until the end here, this would be the last base of that chromosome. And the boundaries represent that you are changing chromosomes. This is a new way of looking at DNA. And then we have this. So this is really cool. So it's for people that are used to look at high C maps, this is really cool. So this is Urtica dioica, and it's a tetraploid species. So basically here in this diagonal, the curators have curated all 4 haplotypes. So basically a polyploid. So for humans, we have 1 chromosome for the mom, 1 for the dad. When you have 4 chromosomes, you have duplications. You have 2, like an extra pair for the same chromosome, right? For, like in this case, 4 haplotypes. And then, so if we look at this figure here, it's like this is chromosome 1, version 2 of chromosome 1, version 3, and version 4. And we can see that because of the off-diagonal collinearity that you can see in the map, right? So they just put them one after the other. And then you can basically see that they share lots of similarity with the next one. So, and they did that for all of the chromosomes of that species. And this is really nice because now we can even start studying like haplotype biology. Because, for example, here we have the same sequence, but there is a lot of differences, right? So we know that like for mom and dad, we already have differences. If you have 4 haplotypes, you have more difference. So one thing that we can clearly see is that one is larger than the other. So there are insertions, inversions, and lots of different things. So this is really cool. So we can do the very large. So we have then the largest genome ever sequence, which is 94 gigabases. If you consider a human genome is 3 gigabases, so it's 3 billion bases. And here's like 94 billion bases. And it's really nice because now, so this is not tetraploid, it's a diploid species. And it's basically full of heterotransposons. So to assemble it was very hard, and it breaks all the pipelines. It doesn't run automatically. And now to analyze it is even harder because now like 1 chromosome is like a whole human genome. So it will take a while for the science to come out of this, but it's coming out. And we are like adapting pipelines for it. We can do the very small as well. So there is this protocol called PIMS, developed by Chris Laumar. He's now at the Natural History Museum, which can take very like nano animals that are very small, that have very little DNA, and extract DNA from one individual. So the process in the lab, it takes like 3 days for each sample that you sequence it. They are optimizing, but it's still very good, already very good. One example is this nematode, which is very, very tiny, and it has around 1,000 cells per animal. Okay, so it's a very, very small worm. And Erna has used the PIMS method to do her assembly. So she basically extracted, for the contigs, she extracted with the PIMS method from one individual, one worm, and then she pulled lots of individuals to do the high C, and she got to a very, very nice genome. That's very cool as well. So we do more than 1 genome for the price of 1 in a way, because again, because we have very long and high quality reads, if you have contamination or what we call cobionts in the sample, many times we can assemble the entire sequence of the bacteria that comes with. So in this case, we assemble the moth genome, 3 Wolbachia, and the mitochondria. So all in with one sample, which was also very nice. Right. So here, again, you have this presentation is free. We have lots of different portals of all of this information I'm telling you. So, for example, raw data in assembly is in this DToL QC page. So all the species that I'm showing, the species that are in progress, all the high C maps and everything is all there. It's all public. So everyone can see this. All the species that are already annotated by the Ensemble people are in the Ensemble annotation browser. And we have other types of databases. So this one is nice, GOAT, because when you are looking for a genome, you go to NCBI usually, and you type the name of the species. But if someone is like finishing the genome project, but hasn't put it public yet, you don't find it. With GOAT, we have one curator, especially that talks to everyone in the world and tries to get lists of things that are being done and sequenced and uploads it to GOAT. So you can understand, and instead of like duplicating an effort, try to collaborate and see if you find that a species is in progress or not. So GOAT is a very nice tool to have a look at metadata. Our genomes are in the Welcome Open Research portal. All of the genome nodes that have been produced. So far we have 673, a little bit far away from 70,000, but we're going to get there. This is another initiative, which I'm very excited about, which is called Genome After Party, and I'm involved with that as well, but it's mainly Maggio's team's task. So in biology, as soon as we sequence a genome, there is lots of standard analysis that we want to have, and everyone makes them. It's like BOOSCO analysis, you want to see the CG content per chromosome, you want to see the read coverage, like all the reads mapped back to the assembly. And then you want to do variant calls of this, like seeing the different SNPs, right? To see like the deployed differences and etc. We are creating a website that for all of the DToL genomes, the Darwin Tree of Life, and other genomes as well, we are going to have all of these runs already done. So there are a few things. So we standardize the runs. We also try to save in energy, right? Because otherwise people are just computing all of this analysis over and over again without the need of doing that. And this is like, so this is really nice. So Maggio is doing that with his team, and he's also welcoming collaboration. People that want to develop software and pipelines for this are welcome to join. Right, so here at the moment, as 7th of March, actually it's been more than a month now, we have in process 5,446 species. We have completed 2,316 genomes, and we are working in more than 3,500 species and lots of cobionts as well. So not only eukaryotes, but the cobionts are coming out as well. It's all open, available. I'll show you very quickly some science that is coming out of it, apart from the ones that I already mentioned. So this is my high five very quickly. So I was there assembling lots of genomes and finding all the mitochondrial genomes. And then I basically needed to automate a way to get the mitochondrial genomes out. So the mitochondria is circular, right, in many animals. So what happens is when algorithms are trying to assemble the mitochondria, they assemble it redundantly more than once because they find overlaps, but then they never finish finding overlaps. So if a mitochondria is like 18,000 base pairs long, sometimes the assemblers are going to assemble like 30,000 base pairs because there is a redundancy in the assembly. So basically develop a pipeline together with João, my PhD student shared with Mauro. We developed this pipeline to basically take these contigs, remove the redundancy, annotate, and then have only one circular mitochondrial genome. And this is used by the Tree of Life all the time. And now we have pipelines for mitochondria of plants and chloroplasts because they are much larger. They have repeats. They have different versions of the mitochondria. Change is working on this. This is already available on GitHub, but it's not published yet. So it's called OTK. Basically, the idea here is just to take a larger k-mer to do overlaps, and then you just go across repeats easier, and then the organelles come out. This I showed you, the Sicilian work. So this is another thing that I do, which is basically like trying to understand ancestor karyotypes. So if you have lots of mollusks, of like cephalopods, bivalves, gastropods, they have different numbers of chromosomes, okay? And then I'm trying to understand how was the minimal karyotype of a mollusk before. So this is nice because now we have chromosome level genomes, because without chromosomes, we cannot do this analysis. But then you can basically try to understand, so if how the major transitions of life and the appearance of species are related to the karyotype, like what are the boundaries? So what are the chromosomes that every mollusk has? When the chromosomes break, why do they break? What is in that region? Is a transposon? They're like a very like repetitive sequence. So we can really try to understand the speciation and the appearance of life in terms of the molecular biology of the species. This is, we also do biological invasions again. So this is Juan, our shared PhD student. So Juan works with the golden massa, which was also my PhD, but now Juan just destroyed my PhD because he resequenced it with long reads. And he is also working with corals that are invasive in Brazil, which are triploid. So he has been having a lot of fun working with triploid species. If you want to know more about the Darwin tree of life, there is a series of papers explaining why sequence all the eukaryotes and how we are doing this on PNAS. The other thing is that we take, we have a priority list for sequencing. Lots of species that happen in Britain and Ireland happen in Europe as well, right? Because we are like integrated with the world. If there is a species that you're working with and would like to be sequenced, you can fill this form and we'll try to do our best to put it up in the priority list. Almost finishing. And I just want to mention this as well. So together with Anne McCarthy and Sadie Pines, I am a co-chair of the Justice, Equity, Diversity, and Inclusion Committee of the Earth BioGenomeProject. So there are many things together with these amazing people. They are part of our committee as well. So there are many things that we can discuss in science, like women in science, and like so the gender gap, and so there are different Jedi issues, right, in science that we can discuss. But in terms of the Earth Biogenome Project and sequencing everything, a big thing is to avoid neocolonization, right? I will explain. So basically, like, lots of the biodiversity in this planet that we want to sequence are in the global south, which have less infrastructure and funds to sequence their genomes, right? The idea here is that we are this committee to guide and help people when they are making collaborations. So we want to foster the development of many different countries rather than just extracting samples and sequencing them in Europe or in the United States. So we are together to advise on relationships between different parts of the world. So that's the part that I'm most passionate about, Jedi, in the Earth Biogenome Project. If you work with biodiversity genomics, I want to invite you for this conference, which is from 28th of October to 1st of November. It's free and open for all. It's called Biodiversity Genomics. So this conference was supposed to have happened at the Sanger, but then the COVID happened, and then it became online. And then we kept it online. I know that online conferences are a bit annoying, but they are, first of all, environmentally friendly. And second of all, if you don't have the opportunity to travel, which many times we don't have, you have the opportunity to present for a very nice audience and discuss with lots of different scientists in this online conference. So if you have like work in genomics, you should submit an abstract to that conference. The other thing that we have is this Biodiversity Genomics Academy, which has lots of different courses. This is going to happen again in October. And we have already lots of different courses in genomics online. Right, so finishing, I just wanted to present you the project. I hope you have liked it. So we are producing outstanding genomic data for the study of biology and evolution, developing different databases as we go along with the project, developing methods, wet lab methods and bioinformatics methods as well. This is a free information for the community and free, very valuable information to understand biodiversity and the time of the Anthropocene. So thank you so much. Grazie mille. And as let me just finish. One more sentence. So as my boss says, it takes a village, and it's basically a village. So it's a lot of people involved. It's not only me. So I'm very honored to be here presenting the work of many people. This is us. So this especially Shane, which has been my boss until December, and Mark, and you. Thank you so much. Now to that.
Grazie Marcela. Any questions? Qualche domanda? Non ve ne andate di qua senza domande, per cui piacciamo bene.
Passiamo all'italiano.
Quale specie vorreste sequenziare? Che ti interessa? Giorgio? One second. Stand up, stand up.
Volevo chiedere quali altre specie avete già in programma di sequenziazione?
Oh, multi. So many. So if you look at this, yeah, so this one is the one that we have sequenced, right? But so yeah, so we have lots of, so these ones are the ones already done. So that's an interesting question, right? I should have plotted a graph of the species in progress. Okay, but I can tell you, so lots of plants, lots of fungi. We have lots of polyploid things now because we were avoiding the big ones at the beginning, but now we are sequencing them. Lots of animals, and also single cell protists as well. So very small ones, they are all in the pipeline. So the DToL QC page, there you can consult which ones are under sequence, but there's like 5,000.
Thank you.
Why just the named ones, and what's to do about the non-named species?
Yeah, that's something that we will get there when we get there. No, I guess. So first, because Britain and Ireland have a very good history of natural historians, right? People that have described species like Darwin, for example. But we are arriving in places when the description by a taxonomist is different than what the molecular biology shows. So I think we will need to be case by case, first of all. So, and you also need to discuss and respect traditional knowledge a bit. Because people, so for a long time, let's say, two species look exactly the same. And then you sequence them, and they have, the genomes are very different. And then you try to do experiments in the lab, or you know by the ecology they don't reproduce. Or vice versa, right? Sometimes they do reproduce, but they look very different. And then you try to understand why. So it is a case by case at the moment. So we are, so the answer is, because we have plenty of work already. That's not it.
So I have a second question, which is really trickier. You're showing a lot of protocols that you're trying to standardize, but I haven't seen yet one programmer or coder that accepts the code of someone else. So that, how do you think, you know, everyone wants to do their own analysis and they have a reason that makes all sense, at least in their heads, why to do their own analysis and run in their own computer and then do everything. So how do you see this and the effort? Is it worth the effort to try to standardize all these if, you know, people want to do what they want to do?
It is worth it because a little bit of variation you are going to have because biology is in the way, right? But for example, this paper here is is the Vertebrate Genomes Project pipeline that we put it on the galaxy, you know? So if you have PacBio reads, there are a few programs you should try. It's people sometimes they want to take slightly different paths because depends on the amount of repeats a genome has, then one thing will perform better than the other. So the thing is, but it's just like it's a confined amount of different softwares you should try. Okay, you shouldn't reinvent the wheel all the time. But yeah, so the first thing you always need to do is to plot a K-mer plot of your genome, because then you can try to understand what's going on in there.
It's more like good practices than good.
Exactly. Yeah. So, and this on the Galaxy has many different setups you can have. And so, and at the Sanger, we are doing a next flow pipeline, which is just basically you can take it from there and put it in your computer, and then you run it. So best practices than protocols, yes.
Other questions? Altre domande?
Which species the sequence first? Like, where do you start?
Yeah, that's a very good question. So first, COVID determined that because the project started and we had lots of lepidopterans, and then we sequenced them because it was easier to collect. Now, yeah, so the idea is to sequence one representative of each family first. But at the same time, when you want to answer a biological question, it's good that you have very close related species sequence as well. So it's a little bit of both. So there are a few projects that talk to us, and then we also have internal projects, and then we kind of guide the sequencing towards these ones. But then if people come from outside and want to establish a collaboration, then we can put them in the priority. But it's basically project-based, and also trying to get at least one for each family in the first few years.
How long is estimated to take for the 70,000 species?
10 years.
10 years.
So at the beginning, so Sina and I, which was the other bioinformatician, we were the first ones in the assembly team. And in 3 years, we did 270 genomes each. Now we have DToL, which is automated, and it has an 800 in genomes in 6 months. So you just need to automate a lot, right? The hardest is the lab. So now the bottleneck is collecting. So some species are harder to collect or to find, and then have good samples for species that are under threat, because you don't want to kill species for genome sequencing. And lab, automating the lab is the hardest part.
Adesso ho una domanda molto interessante per la mia classe che sta affrontando questa tematica. Adesso te la faccio in italiano, poi magari te la rifaccio in inglese. Tu hai detto che con l'esempio dei molluschi possiamo studiare il processo di speciazione attraverso l'analisi della struttura di un genoma, e in particolare dei cromosomi. Per cui tu ritieni che ci sia una sorta di evolutionary constraint which is driven by chromosome structure.
It's a hypothesis. Huh?
And do we have any evidence beyond the mollusk example in this experience of tree of life sequencing?
Well, the evidence is, if you look at extant species that are alive today, the chromosomes, they are conserved, right? Basically, because when you pass on the information to the progeny, you need to be able to divide this material well and pass it on to the germline and produce viable offspring. Because of that, chromosomes are conserved, right? But the major transitions of life, we see lots of different karyotypes. So for cephalopods, like octopuses, their genomes are very shuffled, right? So, but there is still some conservation to others. So what we are trying to understand is what is the minimal viable product, let's say. And when the synteny breaks, so Yeah, what is the minimal conservation a chromosome has to have a species done, right? And then how much, then in these transitions, how much is related to like a heterotransposon going there and changing and cutting, and how is this cutting and paste is happening across evolution? So the evidence that we have that conservation is needed is that we see it, no?
Okay. So this will next step in our class. Please.
Io volevo dire, se le specie a rischio di estinzione hanno la priorità sul potenziamento.
Sì. Sì, sì, sì.
Come siete messi con il
Per i dei
avete
difficili.
già sequenziato i dischi oppure siete cioè state
Quante specie invasive?
I don't know. Sorry. Invasive, I don't know. It's a good question. Rischio di estinzione? That's a good question. You ask very good questions. I need to plot all of that. I don't know. But many.
So we don't follow the IUCN list, or that is not on the.
We follow, but it is based on accessibility. The project needs to happen, right? So these decisions are taken every few months. Like, let's put those on the list now because we have samples. So this is the thing. So if you have a partner that has a sample, then they send it to us, it's easier for us because we need to collect all the samples. So it varies. It's like a project related, you know, partnership related. Yeah.
Okay.
How do you take samples from bigger animals? I know with worms you put them in the plotter in potter, but with bigger animals or mammalians, how do you take samples?
Yeah, so I am not a field scientist, but that's not a good answer. So it always goes with experience, right? So people decide all of that prior to going to the field. So you always, it's the same with genome sequencing. It's like you try to know, it's okay, for a mammalian sample, what is the tissue that gives me most, like, if you're trying to get mitochondria, what is the one that has lots of mitochondria expressed, but doesn't have, I don't know, something that comes from the intestines or some type of protein that would make my sample very dirty, no? So it's very species based, and you need to talk to the specialist and ask like, oh, we already extracted from blood, and blood's not very good. So we extract from this for mammals, and then from bloods for birds. And it's really decided on the pre-knowledge you have from the species, I guess. And if we don't, then what we do is that we extract more than one tissue, or like we store more than one tissue, and then we try with the first best guess, and then if it doesn't yield very good DNA, then we go for the next tissue.
Va bene, grazie allora. Grazie Marcela, è stata
Grazie.
una presentazione molto interessante, grazie a tutti voi per essere venuti qua oggi. Okay, there are no questions?
No.
Ciao Marco.
Ciao Marco.
Thank you. Bye bye.